Tēnā koutou katoa, ngā mihi nui tēnei rā. Welcome to our third live stream where we are joined by Peter Buchanan and Marge, who are both fungi scientists here. They study fungi and they're going to show us a tour of the New Zealand Fungarium. So thank you so much for joining us. Same sort of system applies. If you have any questions, please type them as comments and we will ask them throughout the live stream and also follow up with any questions afterwards. So thank you so much for joining us, both of you.
Great. Thanks, Steph. So we begin here with the po. This is a Maori carving which includes fungi. Now, it is the only Maori carving that includes fungi, and we actually commissioned this from Ngāti Whātua. So their master carver, Dennis Conway, carved this many years ago, and you will see here the mushrooms that are actually, you may well have seen them before. They're actually a blue mushroom, and they're on the $50 banknote that we'll talk about a bit later. Down here, there is a red, well, we might call it a flower fungus that's here. It actually is a stinkhorn. It smells really bad, and it disperses its spores by attracting flies. And down here, there's a little cup fungi as well. So 3 different kinds of fungi on this very special po with important carving that Dennis actually has used in fill carving with meanings for Ngāti Whātua's understanding.
Great, so shall we make our way into the Fungarium down the hallway?
Sure. And we dedicate the area to Dr. Cunningham, who started this whole thing back in the 1920s. And we'll see a bit more about him later.
Cool.
Not too much.
So how old is the Fungarium?
About in the 1920s, Dr. Cunningham started it, and basically the material that he accumulated, many of it as old as 1850, is still present and in great condition today.
Okay. And as we go into the Fungarium, there's a special mat. Do you want to tell us a little bit about why that's there, Marge?
It just helps keep stuff that you might have on your feet on the mat instead of dragging dust and spores into the fungarium. So just we want to protect our collections against the insects and other spores that we might bring in. So
yeah, so it's really protected, everything in here, temperature controlled. There's no artificial, it's all artificial light.
Yes, it's all artificial light. So this vault was actually specially constructed for the collections. And so it is, temperature control is about 18 degrees centigrade and about 40 to 50% humidity. And anything that's brought in here, all of our fungi have been dried at 45 degrees. And then they are put into the -20 freezer for 7 days before they're brought here. And that just helps protect it against mold, mildew, and insects that might hatch during the process of drying them. So,
yeah. So there's about 100,000 specimens in here?
Yeah, over 100,000 now. So it's the national collection of fungi, and it's, as I say, been going since 1920, and we have about 2,000 additional per year. Yeah. So we're trying to document all of the fungi of New Zealand. We're a long way from doing that because maybe only 2/3 of the fungi of New Zealand, only 1/3 of the fungi of New Zealand are known currently. We've got a long way to go, but this is the national collection where we've got a great representation of the native fungi and also the introduced. But Marge is the curator, sorry.
Yeah, so we have, we know about 8,000 species of fungi that are present in New Zealand, and so we estimate that there are probably about 25,000 species altogether. So there's quite a lot of work to be done. So, I'd like to tell my students that if you ever want to get out of forgetting Mother's Day, you need to find a new fungus and name it after your mother, and then that way you'll be set.
Yes. So shall we have a look at, you've talked about the drying process, everything in here is dried, and it's also frozen. Shall we have a little look at the freezer and that sort of process?
Yeah.
Take us down there.
Yep.
And so as we walk down, obviously there's boxes and boxes and cabinets
Rows
and cabinets.
yep. It's like a museum, but everything's a bit more enclosed than a typical museum. You also might notice that all the walls and floor and ceiling are all concrete, because we want to have an environment that we can maintain with the least amount of energy. We also got a gas flood fire protection system, so we don't allow water in here at all. So they're the pipes that are upstairs. But have a look inside the freezer.
Okay.
So this is a -20 This is a -20 freezer, and so anything that comes into here, any of our specimens have to be frozen here for 7 days, and then they're brought in directly. So that's why the freezer door opens. The freezer door opens into the Fungarium directly, so you don't even have to walk out in the hallways to bring the frozen specimens in.
So they get into there initially, coming from the corridor outside where our offices and labs are, into there, and then this other door is their entrance into this collection.
Okay, so it's a second layer of protection.
Absolutely,
Yeah.
absolutely, yeah. And then if there is any, if we think that occasionally you get some insects, if we have found anything, because there are these type of book lice that really like to eat fungal spores, and if we do find them, then all of those boxes go back into the freezer, and then they're brought back again, and that just helps us to control the insects. And the really nice thing about that is the fact that initially what they used to do to control the insects was to fumigate them with harsh chemicals, and so we wanted to prevent that, and this is a really good way of doing it.
So it's very cold. How
Yes.
cold is the freezer to kill these nasty little insects?
It's -20 degrees centigrade.
Okay, -20.
So it's really just it's a domestic freezer on a large scale. Yeah. We won't close you in. Right.
And so you've taken out a few specimens down the aisles. Let's have a little look at what
Yeah. Well, this aisle here, most of the fungi that we have in here are associated with plants, because fungi are, they have relationships with plants and with animals, but we often see fungi affecting plants, and so this collection contains a whole lot of fungi that are damaging to plants or are found on native plants without causing any problem at all, often decomposing the wood or the leaves. But there are some that actually also interact with animals, and there's a whole range of them. We don't have that many specimens of them, but the one called awheto, in Maori awheto, or vegetable caterpillar, is one of the more dramatic ones, I guess. And what you see there is a dead larva of a moth, a native moth, and that was underground when this fungus infected it, or maybe it was feeding on some leaves, and then it actually started feeling rather sick, because what happens is the fungal spore grows, and the fungal material, growth form is like tiny threads called hyphae. These hyphae actually start to grow inside the caterpillar, and they have actually fed on the inside of the caterpillar, obviously killing the caterpillar, and the caterpillar withdraws into a burrow in the soil. And when it withdraws into the burrow, it always has its head uppermost. And once the fungus is fed on the inside of the caterpillar, and the caterpillar is obviously dead, the fungus needs to get above ground so it can produce its spores. And it produces this structure here, and ground level probably was about there. So this is what sticks above the ground. Normally only one. I don't know why this is two. And it produces its spores in these structures here, which, if you could look closely, are tiny, tiny flasks, a whole lot of them, and the spores are formed inside those. They get shot out into the wind currents, and that's how it's dispersed. So this was a fascinating fungus just because of its biology, but also fascinating because Maori collected this and treasured it, because they found somehow that If they collected these and they burnt them, they could produce a charcoal, which when it was powdered and mixed with bird fat, gave a very strong and long-lasting black pigment, which they used for tamoko, for the tattooing of the face and other parts of the body. So very strange to think of a fungus-insect combination called a whetau, which was used for pigments.
And also having so much of it underground, it could so easily be missed.
Well, all you see, yes, above ground is just that. And so I have trouble finding them. And I found 3 in one day and I was over the moon. But obviously Maori know how to find them. And maybe they were more common back then because they're not, I think, not so often collected now.
And so what we have is the boxes that all of the fungi are in. Marge, could you talk me through a little bit of how they are stored and why they are in these packages and the labelling process and things?
So part of it is just we want to capture the information about the specimen. So we want to know what it is, of course. So if we know the name, so here you have Cordyceps robertsii. That's the person who named this fungus, and not the person who collected it and determined what it was, where it was found, and that's really really useful information because that just helps you be able to find things again, to know what the distribution of that particular species is. So this was collected by 2 people and then we have the date of the collection, and that again helps us because it gives us information of when we might be able to see this fungus, like what is its range. It's because fungi are, even though the fungus could be present year round, often the fruiting body or the mushroom is only produced at a certain a really short time frame, so that we want to know like when it's actually when it's found, and most often for mushrooms, they usually April, May, June is kind of the best time to be able to find mushrooms. And then over here is the person who identified it, so it's one of the collectors, and he identified this as the Cordyceps robertsii. So as we mentioned before, everything has been dried. And we put them in packets just because you can store a lot more in a box with it. All of the paper is acid-free paper, and that just helps preserve the collections, because what we want to do is to be able to keep these for generations. As Peter mentioned, and we can show you one of our earliest collections is from the 1850s, so it's a really long time ago. We want to make sure that this collection is preserved for future generations. So we use best practices, including having this acid-free paper to protect the specimens.
Should we go have a look at some of those?
Oh, can I show you one more?
Yeah, sure.
Another of our sort of special insect fungi is this one here, and this is on a weta. Now you see the legs of the weta are actually fallen off. So a dead weta, and those little sort of structures over the body of the weta is a fungus that may or may not have killed the weta. We don't know yet. We don't understand the biology of it, but it's a native fungus on a native insect, so the chances are that they're in balance and there's no sort of damage to the population. So a natural process, possibly actually just growing on dead weta for all we know. Or if it is actually a parasite, then it's not a parasite that's going to damage the weta population.
So that remains unknown.
It's work to be done.
What killed what?
Well, absolutely. There's lots and lots of questions like that. We've got lots to do.
And so we're going to have a look at some of the specimens that you've taken out.
Yeah. Show.
Because when they're dried, obviously they lose their colour. So
yes, they
the
do.
charts that you have up show what they used to be like, but
yeah,
in comparison they're quite dull now.
These are beautiful, and there's lots of beautiful fungi if you just go out into the forest, especially around the, as Marge was saying, the autumn time, the April, May, June time. But you dry these and the colour just goes, and they shrink and they distort, which you might say is a real pity, and it is, but the actual essential details that we need to identify them are still present microscopically, so we can rehydrate a small amount of these even when they're dried, and they'll actually swell back to their original size. And the DNA, which is also really important, is present in the dried material as long as it's not been dried for too long or wasn't dried at too high a temperature. As Marge said, it dries at 45 degrees. It's really good. The DNA is still intact and it can be extracted. So we need the microscopic features and we need the DNA. We don't so much need the actual shape.
Okay.
Yeah.
Let's have a little look at what we've got.
So back here we've got a few things out. Maybe we start with the large because I'll go over this side, because fungi are very, very small, and most people almost expect fungi to be small. But this one, for example, is a relatively large. It's not the largest by any means. They grow bigger than this, but this is Ganoderma. It's a wood decay fungus. You can see here, this is where it was attached to a standing tree, probably a standing dead tree. And it has grown annually out. So these are like growth rings of when you cut a tree across, you'll see growth rings in the wood. These are fungal growth rings, and this is about 20 years old. There's about 20 different layers or ridges that you can see there. Each year it expands outwards, and each year it expands downwards. And unlike the sort of conventional mushroom, which has gills on the underside, these ones have pores. And if you could look closely at this, you'd see a whole lot of the mouths of thousands of tubes which go up into this fruiting body. And it's within those tubes that the spores are formed. So when this fungus was on the tree, it was in a position so that those tubes were exactly vertical, and only in that way could the spores that are produced fall out of the tubes and into the wind currents. So really fundamental for this group of fungi especially to have those tubes exactly vertical so that this grows horizontal, the tubes are then vertical. Now, this is more solid than the wood it grows on. It's really heavy, it's robust, 20 years old at least, before it was taken off the tree, and it's solid. Similar size, but a quite different structure, is this one here. This is Laetiporus, and it is, as you can see, I'm just holding it with one hand, it's very light. In fact, it's weighs about what you'd expect if it was made of polystyrene. And that gives a clue to how fast it grows. This 20 years, this one maybe 3 months. It grows rapidly and it only lasts for a short period of time. It grows on a living tree, so it is actually a parasite, but again it's a native parasite. It isn't damaging the host, and in fact the host that this grows on continues growing, but it does feed on part of that host to produce this large structure. But it doesn't have the density of this other one. On the underside, again, you've got these tubes and the mouths of the tubes. They're actually larger this time, so you might be able to see those as actually holes. And those tubes, again, are vertical, and the spores are formed inside, and they are shot out into microscopically, they're shot out into the middle of the tube and down, and then into the air currents. So this one actually falls off its tree. You can see the top area here is sort of a bit fragmented, and that's because when this upper surface breaks down at about maybe the 3-month time, water then gets in and it becomes too heavy to actually stay on the tree. It was only attached by this part here, and so once it gets soaked by the water, instead of the water running off, it falls to the ground. Now, the importance of that, and the reason I've explained that, is because Maori used to, and still do, collect this fungus from the base of the tree that it grows on. And they collect it because once it is dried in this form, you can break a piece off and you can use it as a way of carrying fire. And it's amazing. It does break relatively easily. I won't do that to this specimen, obviously. But if you put a match to it, it will form a small flame. You can then just blow it out and then later blow on it again, and it will come back as fire. So useful for carrying fire for up to a day or two, I understand. It was also sent, parts of these were sent to soldiers in World War I to light cigarettes in the trenches. So a culturally important fungus, another culturally important fungus. Very different consistency from this one. So these are the larger fungi.
Don't forget about this one.
Oh, well, there's another
one.
large one.
So this probably, Peter told you about the one that took 3 months. This probably grew in maybe a couple of weeks or so. It's a giant puffball, and most kids are familiar with it because when you find it, then it gets kicked around like a football, and it's super, super light. But it did weigh 8 kgs, as it says over here when it was first found. But now it's like, just maybe a couple of 100 grams or so, less than a kg, definitely. But you can see as well, it's just a packet of spores. Like this is what the mushrooms are. Is they just have tons and tons of spores in them to be able to disperse and land in places that they can grow again. So.
And puffballs like this, they're white when they're young, and they're white inside when they're young too. But this one that Marge has got, it's fully mature. It's just hundreds of millions of spores, I think we could conclude in there. And that brown dust is just, if we opened the packet, it would be everywhere.
So we're not going to open
We're not going to open the packet, no.
Cool, put it away.
Yep.
So, Peter, why is it so important that we study fungi?
How long have you got? Well, as Ruud Kleinpaas said at the launch of a book last night, he said fungi rule the world. And they kind of do. They're present in all of the environments that we go to. They're present in the city, they're present in the forest, they're present in farmland, but they're often not seen. And what they're doing, though, even though they're not seen, they can be doing all sorts of important things. Like almost all plants need fungi with their roots to grow as well as they can. And plants and fungi have had this association, this beneficial association for hundreds of millions of years, ever since plants came from the sea onto the land. Because when they came onto the land, actually the fungi were already there. So they took up relationships with organisms that were already on the land, and the fungi were the largest organisms on the land then, apart from maybe some animals. But in that area, the fungi were actually often the size of trees. These prehistoric fungi are very different to what we have today. Anyway, fungi and plants linked up, and they've been that way ever since. So the beneficial role of fungi in plant growth, really important with the roots. The whole decomposition process, so when plants die, or when branches are blown off trees or leaves fall during, if they're deciduous, something needs to get the nutrients that are in those fallen leaves or branches or trees back into the ground to give nutrients that other plants and other animals and other organisms, including fungi, can use. So that decomposition process is driven by fungi. They're the main agents of decomposition. If we didn't have them, we'd have the place absolutely full of dead plant material as things mature and die. That decomposition process is fundamental. So the beneficial with the roots, the decomposition, and then there is a parasitic role, which sometimes is good, sometimes is bad. We've got a few things, if I scratched my head, you might not realise that it could be actually a fungus that's actually resulting in the dandruff that I haven't got right now, but I have had in the past, in my hair. If you looked at my toes, which I don't advise you to do, as my family knows, I have some fungal souvenirs on my toes. So fungi are all around us, and inside us too, but we actually control the fungi pretty well. We naturally are not prone to problems with fungi in any great way, apart from these sort of more cosmetic things, tinea and athlete's foot and those sort of things. And then parasitic fungi, in terms of their parasites of plants, yes, some of our crops are really, really prone to diseases, and most of those diseases are caused by fungi. But you probably don't have sympathy for fungi doing damaging things to plants, but what humans have done is they've got all these plants and they've put them all in rows together. Now, once a fungus has actually grown on a plant, it needs to get to another plant to be able to spread itself, and humans go and plant all these plants together. So it's really easy for fungi to just go from one plant to the next, to the next, to the next, because we plant things in what's called monocultures. Monocultures are great for fungi. So then we've got to use fungicides to control the fungi that are present on these monocultures. So there's a whole area of plant diseases that we also work on. As well as valuing the native fungi of New Zealand, we also work a lot on introduced fungi on our crop plants.
And the fungi come in all shapes and sizes. We've got some really large ones and some really small ones, and they're all weird and wonderful. Can you show us a little bit more about what we've got around here? Marge, potentially talk us through some of them?
All right. So one of the things that I study are rust fungi, and so those are the plant diseases that Peter was talking about. This is one that's a native rust. It's Puccinia otagensis. It's on clematis, and you can see the flowers of the clematis vine. And what this fungus does, you can see the pustules, so these yellow spots on it, are just, again, lots and lots of spores on there. And what it does is actually distorts the way that this vine grows. So it almost looks like a snake. And this was one of the earliest, one of our earliest named fungi in New Zealand. But the interesting thing about this is that it actually needs two different plants to complete its life cycle. So it can grow on here, but in order to complete its life cycle, it goes on to Muhlenbeckia, which is completely different and completely unrelated to the clematis. So, and it looks really, really different as well. But this is the same fungus on two different plants. So, that's one of our things. And then
there's a number of fungi. I mentioned already the one that Maori used for fire carrying. There's a number of other fungi that are important that Maori value, as especially when obviously they're living in a forest situation. We recently produced this book for Kurakaupapa, which is talking about the Maori knowledge and uses of fungi. And now Hekaheka is a Maori word for fungi in the broad sense. And so another of the fungi that Maori value is this one here, which is known as Hekaheka. This is also known as the wood-ear fungus, and when it's on a plant, when it's fresh, it's actually an ear shape, so it looks very much like an ear, and it's got the texture of an ear as well. And it's rather common, and it was found in the early days. Maori were collecting it for food, and a Chinese entrepreneur of the time in the late 1800s realized that this was also well known in China, and it was collected in vast quantity and dried and exported to China. So such fungi like that are also in this this publication we've done. This is actually in Maori and in English, Te Reo in English. This is the one that we had up there with the fire carrying. This is one that we've got here. So this is armillaria or harare. This is probably the one Maori eat most commonly from the forest. It reproduces it produces its mushrooms in winter time, so it's also known as the winter mushroom. Quite widespread in New Zealand.
Quite commonly found.
Yes, very common, especially in winter on fallen logs. Yeah. This one here is it's a parasite, a native parasite. It's quite a large mushroom, and you can see that it has what we call a veil here, which gets covered by the brown spores, and that sort of hangs down like a skirt, quite diagnostic. This is the wood ear, so this is this one here. So you can see when it's dried, it's lost its colour, and it's been quite deformed in shape. But that's what is quite common in the forest. You may well have seen that. The basket fungus was also eaten in its young form, and we have a specimen of that over there, because this fungus hatches out of an egg, and you see the egg there on the right hand side, and the actual structure that hatches out of that is shaped for some strange reason like a basket. It can actually move potentially, but you often smell it before you see it, because it is one of the stinkhorn fungi, and it has a terrible smell designed to attract flies, and it's the flies that are the dispersal agents of that fungus. So they stink to attract flies. The flies then come, feed on it, the material, and take the spores with them.
And if you do find it, you can put a balloon in it and then dry it, and then it keeps that basket shape. So you then just deflate the balloon afterwards, and then that's how it we got it to keep its shape, because otherwise it would just completely collapse and become like really flat. But you can keep it if you do find it, and they do you do find them very commonly in wood chips, and just in the city you can just go out and find them. But yeah, that's a cool little project to do.
Mm.
And so there are so many species of fungi that are unnamed in New Zealand, and then also again some that are endangered. So talk me through where are we at in history with fungi in New Zealand, with protecting and also finding?
Yeah, well, it's relatively, I think, unknown to the public that there are threatened fungi, just like there are threatened animals, plants, and all kinds of animals. In fact, the focus in New Zealand is on threatened birds, and I guess threatened reptiles, and a little bit of interest in threatened plants amongst the public, I'm saying. But there's also threatened fungi, as every group has its rare and endangered species. And the fungi haven't really been looked at until quite recently in that regard. And yes, we have I'll give you an example of one that's probably one of the most dramatic examples. It's this one here. So this is another bracket fungus. This is the same group, Ganoderma, as this one here. But this Ganoderma is only known from 3 specimens ever. And it's only been collected by one person ever. And that person is now dead. This specimen was collected in the 1970s, 71 or 72. All 3 specimens from 69 to 72, all on Pukatea, all in the Mount Pirongia area in the Waikato, and no one has been able to find it since. It is a mission of mine and a few other people to find this. And we've looked many times, and we've had some public field trips and that sort of thing. We are still looking, and I can't accept that it is extinct. It is possibly extinct, but I can't understand how it could be, but boy, is it hard to find.
And so you're just talking about collecting, and a lot of fungi are still being collected and named, and that's around 2,000 specimens each year coming in?
2,000 specimens.
Yeah.
The rate of naming is nowhere near that much, because naming is a very complicated and detailed process.
Yeah.
So how many new discoveries would you find each year around?
Oh, that's a good question. It's a little bit hard to say, because sometimes like you work in a particular group and then you can name a bunch of different species in there, but maybe I don't know, like like some years there are none, but then some years you could have like 10 or 20 being named, and it just depends upon who's working in which group. So yeah.
I mean, in a good year we might have 100 or more.
Yeah.
But it's not at a huge rate, and it needs to increase. In fact, the whole biodiversity crisis for the globe requires a much increased rate. What we're doing currently with rare fungi, threatened fungi, is actually trying to look at where their status on the global red list. Now, Department of Conservation, we have done an assessment, and we need to update the assessment in New Zealand for the Department of Conservation threat list. But recently, if you get a chance, have a look at this Forest and Bird magazine, which is just the latest one out. And in there is an article called Secret Life of Fungi, and it's actually talking about the workshop that was held in Melbourne at the end of July, where for the first time we spent a week doing actual global assessment of about 70 threatened New Zealand and Australian fungi. And that was done in conjunction with some experts overseas who've done such workshops before, and we are currently progressing them to get official red list status.
We've had a question come through. So the fungi you were talking about with the shiny top, is it Ganoderma?
It is a Ganoderma, yes. Yep. And Ganoderma is common. This one here is common. And people who, initially when people first hear about this rare Ganoderma, they then find a wood decay fungus forming a shelf on a tree or a stump, and they think, I've found it. But I've found those ones also. They're not the ones that we want. And you might say, well, what's the difference between that and this? Actually, it's quite subtle, but you see the underside is pale, and the top side has a hint of shininess. A lot of the shininess will be present under here, but that brown dust that you see over there are brown spores, masses and masses of spores, which was why I don't think this thing could be extinct, because it's producing so many spores. Some of them ended up on top. Hopefully the rest of them are dispersed in the environment, and surely somewhere it's growing. But of course it can be growing, and fungi are present all around us all the time, but until they produce their reproductive stage, we don't see them. So we could also be using DNA maybe with Pukatea to try to find this fungus living in the Pukatea before it actually reproduces.
So what should people do if they find a fungi that they think might be rare or that they want to see what it is? What should people do to help?
I think probably like the best thing for people to do is to go onto iNaturalist. And so, perfect. So iNaturalist is a website in which you can upload your observations about fungi. So you just take a picture of it. With mushrooms, you definitely want to get the underside, so you can look at the structures, the gills or the pores, or and so a really nice photo. And we usually have experts who are monitoring this all the time, and they could probably easily or readily find out if it's something rare or no. And then when they do, then we will eventually get to hear of it. So
it was just 2 days ago, a colleague of ours who's actually based at Lincoln as opposed to us here in Auckland was looking on iNaturalist, and there was a record from Nelson of a mushroom-like fungus that's only been known from 2 collections before, and here was a picture of the third, and that came through iNaturalist. So this is a fantastic website, as Marge said. It's hundreds of thousands of observations of people of all forms of life, fungi, animals, plants. Put up a photo, say where you found it, and wait, and people are looking at it each day. And you will likely get a name for it. And you may, if it's really significant, you may get approached by the experts who want to follow up on that observation you've made.
Let's have a little look at what we've got in the cabinet, because we've got some really special fungi in there, so special that they're behind the glass, that you could talk about quickly and tell us what's in here.
Right, shall I?
Sure, yeah.
You start.
Well, I was going to say, one of the ones that we've already seen that Peter had pointed out was the vegetable caterpillar. And so that's, is one of those things that are just so spectacular when you do see them and find them. But this was our earliest specimen that has been put into the collection from 1850s, and that was collected by Colenso. But you can notice that, so as we had talked about earlier, that the fungi do lose their color as they dry, but some of them can retain it, and this is a collection from the late 1970s, and there's almost the same color, so it hasn't lost its color over there. That's one of the evidence.
Well, there's a number of, just some examples of fungi. These have all been freeze-dried, so they're in slightly better form than if they had been just heat-dried. This one here is one that people know if they have it, or if they don't know they've got it, they might find out and be somewhat concerned. This is the dry rot fungus, and it actually can be really damaging in houses, wooden houses that is, with wooden foundations. And where the basement area or under the house, if the ventilation is not sufficient, you can get very humid, moist conditions and still conditions, and that fungus, because the spores are in the air all the time, can grow on the wood, untreated wood, and on paper, and it can move quite rapidly through the framing and floorboards of houses. And so I've seen examples where that fungus has taken hold, and you need builders to actually repile the house or redo the floorboards, or even if it gets into the wall structure, you may have to completely redo the house. It doesn't affect treated timber because it can't grow. The treatment protects wood from fungal growth. But in older houses where there is that change underneath the house to stop ventilation, dry rot is a major problem.
And what we haven't spoken about is how fungi are becoming more well known, and how we now have one on our $50 note.
That's right, and I have it on. That's right, I forgot. All right, well, and we have a picture of it here too. So here is the blue entaloma, and some years ago the Reserve Bank decided that it would be a really good image for the $50 note to have on it the kakako bird and the blue mushroom. And the reason for that is that it's a tuhoi story, that the kakako bird gets its blue wattle, which is on its cheek, from rubbing that wattle against the blue mushroom. And it's all set in Pureroa forest. So it's a great story that is the basis of the $50 banknote design. And this is the latest design, but the previous design actually had the mushroom in a different position and a different design. The mushroom was down here. And when we heard that the Reserve Bank was going to redesign the note, we wrote to them and said, please don't lose the mushroom, because it looked like it was heading off the side of the note. We had some discussion with the Reserve Bank. It was really nice to actually talk with them. And they were very interested to learn more about the mushroom, and they created this design, which, I'm very happy about. And I used to say that this mushroom, this banknote is the only banknote in the world with a mushroom on it. But I've found out that I've been lying all this time. There is a banknote from Costa Rica that I was told in Melbourne in July, also has mushrooms on it. Much smaller mushrooms, not as significant, not as pretty, doesn't quite rate, but it's still a mushroom. So we have one of the 2 notes in the world with mushrooms on it.
So we've had a couple of questions come through, and obviously this collection is very important for New Zealand and the research that's happening. Mm-hmm. What are some of the projects that are happening and research projects around fungi at the moment?
All right. Well, one of them is concerning a very important disease that Marge is leading a collaborative of nationwide collaborative of researchers on, about the myrtle rust fungus. Yeah. Marge.
Okay, so myrtle rust is a rust, similar to the one that I showed you before, except it's an exotic one that came over that flew over the Tasman, and it is infecting all or native mataceae, so things like Manuka, Kanaka, Aotearoa, and others. And so that's the program is going to be looking at to see what the pathogen is doing in New Zealand and how we can control it, and how our ecosystems are going to how they're going to be impacted by this disease. So that's one of the projects. We also have there are 5 mycologists who are on staff at Manaki Whenua, and so we have our various expertise. So Peter, you might have picked up works on Ganodermas, on wood decay, polypore mushrooms, and then we have Eric McKenzie, who has recently retired, but he still comes in and he works on rust fungi, and he also works on
molds. I guess
yeah,
you call them molds.
Yeah, molds. I guess it is probably
very small fungi.
I was trying to think here. And Peter Johnston, who works on he wouldn't like me saying this, but little black dots on sticks and leaves. So and so we have different people working on different projects all the time. And then we also have a lot of international collaborators who come over here because our fungi are so unique and so different from anywhere else in the world, to look at how these different fungi are evolving from groups that they may be related to.
So they draw on this knowledge base here.
Absolutely, yeah. So we do send out loans of our specimens that have been collected here in the native forests overseas to be looked at and compared to fungi that have been found in other places.
So you might know how to answer this question because it's in relation to your area, Peter. How long would a Ganoderma spore last?
Oh, there are so many questions. I mean, when I sometimes talk about the fact that I've studied fungi for over 40 years, I also say I know about this much about them. I mean, and this is an example of what should be a simple question, well, how long does a spore last? And I don't know. I could guess. Because the Ganoderma spore is brown and it's thick-walled. I think maybe it lasts a couple of years, but I don't know. And no one would have studied that for New Zealand, as far as I know, although it would be neat if someone actually came up and said they had. But the fungal kingdom is larger than the plant kingdom. So, Marge said there's 5 people studying this. We've got a staff of 10, say, but there's 5 people studying a kingdom larger than the plant kingdom. I mean, it's a massive, massive group of organisms, and most people sort of just don't even realise it's there often. So much yet to be found out. But that makes it fascinating. I mean, 40 years on and I'm still fascinated about fungi. It's like a disease. and just actually, just last night, actually, this book was launched. Ruud Kleinpaas launched this book at Auckland Museum. This is a wonderful 400-page book about Hauturu, Little Barrier Island, which is New Zealand's most pristine environment. It's fortunate that the island has been protected for so long, and you go there to see New Zealand as it once was. It is very, very special. It's also very hard to get to as a result because they have to protect it. And there's a chapter now on the fungi of Hauturu, and some photos of fungi that have been found on Hauturu, some only on Hauturu, although most are actually also known on the mainland as well. But there's lots more to be found out about the fungi of Hauturu, as there is about every ecosystem, every environment in New Zealand.
Great. Well, thank you so much, Peter and Marge, for your tour of the Fungarium. And thank you, everybody, for tuning in. If you have any more questions, please continue to type them. I know there's been a question about where they can find the resource Ngā Hekaheka or Aotearoa, which is online. Yeah, it is online. It's PDF available, and it's also on Science Learning Hub, which is they've put it there with English and Te Reo together. So yes, it's available electronically. So yeah, that would be a good place to go for it. And if you find any fungi and you want to know what it is or what to do with it, iNaturalist is the best place to go.
Ideal, yes.
And any more information, go to our website, landcareresearch.co.nz. Thank you so much for tuning in. Ka kite anō.
Thank you.
Thank you.
Thank you both.